Hello and welcome to FACT's webinar, examining the ramifications of mRNA vaccines for livestock. Our guest presenter is Dr. Alan Williams. I'm Samantha Gaston, FACT's Humane Farming Program Manager, and I will be moderating this session. Thank you so much for joining us. Before we dive in, let me take a minute or two to do a few quick introductions. Food Animal Concerns Trust, or FACT, is a national nonprofit organization headquartered in Illinois that works to ensure that all food-producing animals are raised in a humane and healthy manner. We accomplish this by supporting humane farmers through our humane farming program, promoting policies that make foods from animals safe and healthy to eat through our Safe and Healthy Foods program, and generally help consumers make informed food choices. Our mentorship program application period closes tonight. So make sure if you're interested in joining our mentorship program that you do so today. Our Fund to Farmer grants are currently open and accepting applications. Please visit our website at foodanimalconcerns.org to learn more about our farmer and consumer services. At this time, I'm very pleased to introduce our esteemed presenter, Dr. Alan Williams. Alan is a sixth-generation family farmer and founding partner of Grass-Fed Insights, LLC, Understanding Ag, LLC, and the Soil Health Academy. He pioneered many of the early regenerative grazing protocols and forage finishing techniques, and now teaches these practices and principles to farmers globally. He is a recovering academic, I love that, having served 15 years on the faculty at Louisiana Tech University and Mississippi State University. He holds a BS and an MS in animal science from Clemson University and a PhD in livestock genetics from LSU. He has authored more than 400 scientific and popular press articles, which is unbelievably impressive, and is an invited speaker at regional, national, and international conferences and symposia. We are very lucky to have him with us today to share his experience and expertise. So without further ado, I will turn the floor over to Alan. But first, I think we have a poll that you would like me to run, right? Alan?
Yes.
Okay. There we go. Sorry, I'm going to pull it up. All right. Can everybody see that okay? Give that a few minutes, and I'm going to stop sharing, Alan, if you want to go ahead and start sharing.
Okay. Well, trying to get it to come up here.
That always seems to be the problem with everybody. We even practiced, and it happened straight away.
Came right up.
I know.
I'm gonna go stop share and try it one more time. And if not, you have them. You can pop them up.
I do. Let me see if I can find them. So you're sharing, it looks like, can you go over to, if you go over to PowerPoint over here on the bottom, click on that maybe?
There we go. Okay.
Then do slideshow.
Okay, are we good?
We did it. We did it.
Excellent. All right.
I'm gonna go ahead and end the poll and share the results.
Okay. Thank you. That's very helpful.
All right.
So we have a large percentage that are direct marketing on the webinar today. I was especially interested in that. Okay, so we are going to look at mRNA vaccine technology and talk about what it is, what it isn't, and then some of the principal questions that we're hearing today from producers all across North America. So okay, now if I can get it to it, there we go. All right. So what is mRNA? Well, it stands for messenger RNA. And basically, this is just a type of RNA that carries a code, okay? A portion of the DNA code to other parts of the cell for processing and making proteins, protein synthesis. So basically, what we find that sections of the DNA code are transcribed into shortened messages that are instructions for how to make proteins. And that's how our bodies and the bodies of our livestock and all of that works, is based on these proteins that are manufactured or constructed in the body. And so these messages through the messenger RNA are transported out of the main part of the cell. And then they, again, once they get into other cells, then they produce specific proteins using these instructions. So it's the foundation for how our cells work, how we work, is really the bottom line. So messenger RNA is essentially an intermediary messenger, okay? So that's what we need to realize and understand. It's intermediary, and one of the things that it does is it's a safety mechanism to prevent outside targeted material from infecting our body and producing foreign proteins. So we need that, and that's a part of what it does. So if the cell recognizes a foreign protein, then it produces very powerful enzymes called RNases that can destroy these foreign proteins. So the messenger RNA gives the cell a way to control our protein production, turning blueprints on or off, so to speak, like turning a light switch on or off. Now, if we want, we can, in the Q&A session, we can get into epigenetics as well and what that means, and I'd be very happy to do that and describe that. But right now we're talking about genetics as a whole. So another thing about messenger RNA instructions is that they are timed to essentially self-destruct. And this is a structural feature of messenger RNA. It has a relatively short shelf life, so to speak. And typically, it's quickly destroyed within minutes or within a few hours, depending upon the types of proteins that are being coded for. This is just a diagram. Everybody knows that, Pfizer and Moderna and others have manufactured mRNA vaccines for COVID-19. And, of course, those have been released now for, I guess pushing 3 years. But this is just a diagrammatic representation of how they do it. We won't get into a lot of detail about this, but essentially what is occurring here is identifying mRNA codes for spike proteins of these viruses, encapsulating them in lipid nanoparticles, and that's how the vaccine is administered, into our muscles in the human body. And of course, the objective is, to create an antibody response to stimulate immunity against the recognized virus cells. Now, in livestock, there are 3 principal types of vaccines. There are 2 that have been used very extensively and for quite a period of time. There is modified live vaccines, And basically, these vaccines contained a weakened or what you might call attenuated form of a live virus. Now, they have been altered to create basically a weakened infection or to mimic an infection, and then to stimulate antibody production and an immune response. A killed vaccine contains an inactivated or killed antigen. Now, these are, because they're killed, they're incapable of replicating in the system, and typically they require boosters to provide, better immune response in the animal. And then the much more recent are the mRNA or messenger RNA vaccines. And these, because they are mRNA, contain genetic material. And of course, the premise is that the messenger RNA tells the body how to produce specific proteins that can create the antibody response against the target pathogen. So let's look at what has actually been happening here. So there has been quite a bit of research over more than a decade now looking at mRNA vaccine development in livestock species. And they actually started developing mRNA vaccine technology in livestock prior to developing mRNA vaccines for humans in response to COVID. This includes researching cattle. There has been research conducted on various university research stations, and there have been collaborating or cooperating beef cattle producers and dairy producers who have used mRNA vaccine prototypes in vaccinating their cattle. So this has been occurring for a little while now. However, in cattle, beef and dairy, currently there is no licensed mRNA vaccine on the market. Approval for any vaccine, including mRNA vaccine, has to go through the USDA. So that's the approving body for livestock vaccines. There are relatively new prescription vaccines, both RNA and mRNA, that have been approved for use in swine, in pigs, specifically for porcine reproductive and respiratory syndrome, or what we know as PRRS. It is currently being considered, and there's research being conducted in developing mRNA vaccines in livestock for African swine fever, avian influenza, and several other food animal diseases. There have been several states that have introduced legislation against mRNA vaccines being legal to use in livestock in those states. Missouri and Tennessee are two of the states that have introduced this legislation. However, no legislation has been passed and gone into effect at this point in time. These vaccines must be kept very cold, and then for use or injection into an animal be warmed and used quickly, or the mRNA will degrade very quickly. So there's issues right now relative, when you look at the research on this, relative to being able to effectively use it in practical situations on farms and ranches. There is also, to make you aware, another type of RNA vaccine that is being developed in this same research called saRNA, and SA stands for self-amplifying RNA. And this allows for higher levels of proteins to be constructed or manufactured in the body of the animal. So the bottom line, as it stands right now, is that There are vaccines that are being actively used in the swine industry that are mRNA and RNA based. There are none in the poultry sector at this point in time, and there are none in the cattle sector at this point in time in the US that are approved by the USDA for use. But again, there has been a decade plus of research moving in that direction. So at some point, there will be approved mRNA vaccines for use in probably every livestock species. So it is coming. The technology, again, to produce mRNA vaccines has been around for quite some time. And bottom line is this, if we look at what they're doing here, okay, in making these mRNA vaccines, they're mixing in modified nucleotides, which are the building blocks of RNA, with unmodified nucleotides. And this basically allows the mRNA to escape detection long enough once it's injected in the target animal's muscle to escape detection long enough to train the immune system for future similar invasions. So that's the premise. And it's packaged typically in these lipid nanoparticles or this fatty outer shell. And that further helps it escape cellular sensors in the body so that it can create an immune response. So that's sort of the background. I know you have lots and lots of questions, so I'm not going to spend any more time on this. I want to get to your questions and address the very specific questions that you have. So, Samantha, I'm going to open it up for Q&A here.
All right, sounds good. Questions have already started coming in. So, which I'm not at all surprised by, because this is always such a cool group of people. So what we have for the very first question, hold on one second. My screen is doing something wonky for me. Okay, so Tammy asks, what is the percentage of herd vaccine associated enhancement respiratory disease in hogs receiving mRNA vaccines?
That's a very good question, and we don't know the answer to that, to be quite honest. The vast majority is obviously being used in the commercial swine sector, the vertically integrated sector. And you know, that's not released from public purview. If you are like us, we do pastured pigs, and I suspect many of you on the webinar do, then you have to check it by law and by USDA regulations. It has to be labeled on the vaccine label on the bottle with, you know, what kind of vaccine it is. If it's modified live, that has to be on the label. If it's a killed vaccine, that has to be on the label. If it's an mRNA vaccine, that has to be on the label. So if you're a pastured pig producer and you are vaccinating your pigs, then you need to check with your veterinarian. And if you do not want to use mRNA vaccines, you need to make your veterinarian aware of that. And you also need to read the labels yourselves to assure that you're not using an mRNA vaccine.
All right. Next question is, do other countries have approved mRNA vaccines? For example, is beef in Brazil that's being imported into America containing mRNA technology?
To our knowledge, no. Now, again, folks, you know, just being very honest, we all know there's things that go on that we may not know for a while. So to our knowledge, no. But does it mean that that's 100% no? It does not.
Okay. Will mRNA vaccines meet USDA organic and grass-fed standards?
So that's another very good question. And it depends on, so, grass-fed standards, there are no USDA grass-fed standards. And I'm not sure if people are aware of that or not, but there's not. If you want an USDA FSIS approved label for grass-fed, then you have to submit a written protocol and affidavits, and then they judge it on a case-by-case basis. Years ago, there was a formal USDA definition within the USDA AMS for grass-fed, but they did away with that. So there no longer is. So you define it, and your label submittal is evaluated for them to determine that. Now, the other thing with the USDA, what you have to understand, particularly with the FSIS, is that grass-fed, if I want to have 100% grass-fed on my label, that has nothing to do with no hormones, no antibiotics, free range, anything like that. Okay. Those are separate approvals. So if I also want to get approved through the USDA, I'm administering no antibiotics, I'm administering no hormones, and they are not being fed in a feedlot, grass or hay or whatever in a feedlot situation. Pasture raised, free range, those are separate claims, and they can be included on the same written protocol. But you can get 100% grass-fed claim and still administer antibiotics, hormones, and feed them in a feedlot. Okay, so that's what we have to understand. All of these, by purview of the USDA FSIS, are separate individual claims.
And Nick put a link, thanks, Nick, to the auditing for grass-fed in the chat for anybody who wants to click on that. So, hi, Alan. Chris here from Puerto Rico. What is your take on vaccines being somewhat of a bait and switch for the previous heavy antibiotic use in the mass production livestock industry? Consumers' pushback seems to play a major role in reducing antibiotics.
Okay, that's a very good question. Vaccine use, first of all, it's not in the US, it's not mandated. So as a livestock producer, I can choose whether to vaccinate at all or not, and I can determine the specific vaccines that I want to use if I'm going to use any. What we do know is this: as you significantly improve soil health parameters, particularly improvement of soil biological profile, and you improve plant species diversity that the livestock are consuming on a routine basis, then you absolutely improve the health status of those animals. And we have quite a few of our clients that are either not vaccinating at all or are using very minimal vaccine. And again, this can be heavily dependent on where you're located, what you're exposed to by surrounding producers that can directly touch your animals through the fence, by the wildlife that you have in your area. So everybody has to, basically make their own decisions relative to the vaccine regimen that they're going to use within their area. I'll tell you what we do, but I'm not saying this is what you need to do. Okay. We do it because we know exactly how our livestock are managed. And I'm specifically talking about cattle now. And we know what our soil health and plant biodiversity status is. The only vaccine we use is a Clostridial vaccine. Many people call that a 7-way blackleg, and that's it. We don't use anything else, but we have pretty much a closed herd, and we have been working at this for a long time. So we feel relatively safe in what we're doing because we don't have the introduction of other things, and our animals are eating a very, very highly diverse diet. I've documented more than 150 different plant species growing in our pastures that our animals are eating from. And we have actually done very detailed phytonutrient analysis, metabolomic analysis on our soils, our plants, and our end products, our beef, our eggs, our pork, our poultry. And we have found that our phytonutrient profile analysis is very, very high. So our animals are quite healthy. We pretty much never need to use antibiotics. So that's not something that is routine and on our farms at all.
Yeah, blackleg is one of those things that always seems to get the healthiest animals if you don't vaccinate against it.
Right.
It's not the sick animals that are getting blackleg. And I think if you could just discuss what mRNA is, because I think there's a lot of confusion as to, so this next question is mRNA naturally occurring, or is it all manufactured? So can you just talk about mRNA's process, what mRNA does in our bodies? And I think that probably will answer that question pretty clearly.
Yeah, so in a nutshell, mRNA has existed since organisms have existed. So it's a natural part of the genetic coding that's in our bodies. So the M part stands for messenger RNA. There's actually different types of RNA, and they perform different functions in coding in genetic coding in our bodies. Our bodies are run off of proteins, protein synthesis, protein manufacture. And basically, messenger RNA or mRNA is the carrier, the mechanism that carries that coding for specific proteins from DNA, into our cells and tells our bodies what types of proteins to synthesize or manufacture. So it is natural, okay, and it always has been. So we've always had mRNA. This is nothing new at all. It's messenger RNA that is in the vaccines is designed, it's a snippet of, the coding from a specific virus that is designed to be injected into the body. And just like any other vaccine, for the body to recognize those foreign bodies and to develop antibodies against it, immune response against it. So that's the premise for the messenger RNA vaccines.
Thank you for that, because I think there's a lot of confusion as to what it actually is, because most people just think of it as part of the vaccine, but it is essential to our bodies producing protein. And I'm going to I just did a quick search, and I haven't watched this video, but there's a video on how mRNA vaccines work. And if you remember back to high school biology, you discussed it then. Some of us are a little bit far away, but I've got a bachelor's in biology, so I remember my college biology of genetics, and I concentrated in genetics. So I remember, not as much as I wish I did, but also, so Chris asks, also, what are your feelings for the needs for vaccines if we're following regenerative processes? I feel like you've already answered that, but if you want to add anything more to that.
Yeah, again, as you move further and further down the regenerative path, then it is absolutely possible to remove most vaccines from your protocol. However, you really need to understand what you're doing. and what your animals can be exposed to and how far advanced you are. We are, on our farms, we're quite advanced, and we can document that. We can document the significant improvement in our soil biology and the function in our soils. And we can document our plant species diversity and the phytonutrient benefits of that. And phytonutrient compounds are more than just what we think of as nutrients. They are also antiparasitic. They are antioxidant. They are antibacterial, antiviral, and they are very, very medicinal in nature. So as we significantly increase that plant species diversity that our animals are able to eat from, and particularly forbs, so Samantha, what a lot of people call weeds, okay? We actually need those in our pastures, all right? Because they are very powerful from a phytonutrient standpoint, an antioxidant standpoint, an antiparasitic, and a medicinal standpoint. I want my livestock eating what a lot of people call weeds every day of their lives. I want them eating that. So I don't want to, quote, eradicate weeds out of my pastures. That's the wrong thing to do if we want truly, truly healthy livestock. So diversity matters is the bottom line. Soil health matters is the bottom line. And again, I'm talking about soil health, not from the standpoint of how many people define it, which is purely soil fertility, a chemical analysis, soil chemistry, okay? That's not what I'm talking about at all. I'm talking about from a soil biology standpoint. That's where you get key immune responses in your animals and establish very strong, healthy immune systems in our animals. So soil biology, plant species diversity, two of the core keys here.
Hey, thank you for that, Ellen. Ed would like to know, what do you qualify as a significant amount of data to be sufficient enough to determine the safety of these new types of vaccines?
So bottom line is there's two key indicators that I look at. One is, do I have a mycorrhizal component in my soils that is at least 10% of my total microbial biomass, of my microbial composition in the soil? So a minimum of 10% mycorrhizal composition, number one. Number two, I want at least several dozen, not several, but several dozen plant species growing in my pastures, and a good mix of grasses, legumes, and forbs. Again, those forbs are critical for that phytonutrient profile growing in my pasture. So I want several dozen of those present before I'm going to consider myself fairly well protected from an immune response standpoint.
So can you just, there's a couple, you're throwing out some terms there that I'm not sure that everybody knows. So you've got microphysical and the phytonutrient or phytonutrients, which could you just explain what those terms mean, just so everybody can follow?
Well, phytonutrients are compounds that exist literally in the thousands in every plant, every animal, every egg, every cut of beef, you name it. They're far more important and far different than what we have been taught to recognize in terms of nutritional profile. If you look at, holding my bottle of kombucha up here, but every food product now and drink is required to have a nutritional label on it, right? But unfortunately, what it gives you on that nutritional label is pathetic. it's calories, total fat, saturated fats, carbs, sugars, proteins. Folks, that's not the sum total of nutrition by any stretch of the imagination. And I can make any junk food by this kind of nutritional label. I can make any junk food look like it's good for me, okay, when it's actually very terrible for me. So that's the sort of we've been taught wrongly here, how to think about nutrition. We need to be thinking a much bigger picture, phytonutrient profile, which again are more than 2,000 different nutritional spectra. But there are major categories like tocopherols, alkaloids, phenols, polyphenols, things like that, which all of those major categories are made up of hundreds of individual phytonutrients. Those are the nutrients that are most important to our ultimate health and the health of our animals and the health of our ecosystems, because contained within the phytonutrients are all the medicinal compounds, all the antiparasitic compounds, the antioxidants that we desperately need, the fatty acids, all of these types of things. So that's what we're talking about when we talk about phytonutrients, not the very broad spectrum of proteins, fats, sugars.
Yeah, thank you, Alan. Appreciate that. Mary would like to know, how much testing does the USDA undertake to detect instances when the vaccine has been adulterated with mRNA by the farmer manufacturer? The manufacturer. Yes, that's right, manufacturer.
That's actually a good question, and that's a question that would be almost impossible to answer, to be quite honest. This is where you take the USDA words for it or not. Bottom line, their statement is that they do extensive testing to make sure that mRNA material is not in a vaccine that's not labeled as an mRNA. Again, that's their statement. Now, to what degree they actually go to absolutely make sure of that, I do not know, and I can't answer it. If there's somebody else on here that can't, a researcher or a veterinarian that can answer that, I welcome their input, but that's pretty difficult. Let me give you an example, okay? We have lots and lots of imported foods that come into the U.S. every single day, right? Including certified organic imported foods. As a matter of fact, the majority of organic foods that the American consumer consumes were not produced here in the U.S. The majority were produced in other countries and imported in. A much smaller percentage were actually produced here in the U.S. in terms of total organic food consumption. But in terms of inspection, as they are imported at ports of entry into the U.S., less than 5% of all foods are actually inspected before they get to the consumer. The USDA doesn't have anywhere close to the number of personnel needed to inspect more than that.
That's a little scary. All right, so Lori says she had to step away for a moment, so she's not sure if this has been asked, and I don't think it has. So do I understand that mRNA vaccines alter the DNA? That concerns me.
Okay, so mRNA vaccines are coding for protein synthesis in the body. Proteins that are mimicking the pathogen that you're trying to establish antibodies against. Now, again, they have a very, very short half-life. And so typically they are eradicated or killed by the body within a short period of time after injection into the body. Now, I am a geneticist. Can I say conclusively that this doesn't escape and get out into the body as a whole? No, I cannot. May it happen? It may. Can I say that it does? No. Can I say that it doesn't? No. We just simply don't have the research, folks. Okay. It's not there. It is not there. And so there's a lot of unknowns here, to be quite honest with you. If it can escape into the body and get into the system, then it's possible that, we are producing brand new proteins throughout the body. That could be possible. But to say that with certainty, no, I cannot.
All right, Casey would like to know, what is your vaccination program on your farm?
Yep, I answered that earlier, but I'll answer it again. We use 7-way Clostridial, and that's about it.
So this person, and let me read through it. It may be that I'll read it out loud. It may be that you've just answered this. It has been reported from at least one university source that COVID mRNA vaccines were tainted, with DNA that could potentially be integrated into the human gene pool. Is this also a concern with livestock? And what are the potential unanticipated consequences of exposing animals to mRNA vaccines? You sort of touched on that.
Yep. So again, mRNA is mRNA. So if it's possible in the human body, absolutely it's possible in the animal's body. There's no difference in that regard. So if you do have issues, then it can mean that your body is now coding for foreign proteins that have not been produced by the body before, and how that impacts us is completely unknown. We do not know how the body will respond, and this will be different in different individuals. This is a very individualistic deal. So we could have a myriad of side effects as a result. But again, there's just so many unknowns here, and we don't have anywhere close to any type of long-term research looking at this to be able to point to. Because again, mRNA vaccine technology is relatively new to begin with, a decade or so. And there just hasn't been, it's impossible to have done the long-term research to detect and determine what kind of side effects can happen. But be aware that with any vaccine, this is not just with mRNA, but with any vaccine, there is the potentiality of harmful side effects.
Good point. Mary Lynn would like to know, can you use mRNA in a specific disease prevention approach, for example, vaccinating against various pneumonia-causing agents?
Yeah, again, so the mRNA vaccine is no different than any other vaccine. It's going to be specific for a specific pathogen, a specific virus, whether it be something like the porcine reproductive and respiratory syndrome, African swine fever, avian influenza, BVD, things like that, in cattle, whatever the case may be. It's going to be designed specifically for a specific pathogen.
And I love this question. Eileen wants to know, what prompted your academic recovery? I think that's a good one. Thank you for that, Eileen.
I don't know that we have long enough to answer that, but I'll give you the very short version. I'm sixth generation farmer and rancher. Family's been on their land since 1840, and always intended to come back to the family farm, but got my bachelor's degree and then got talked to going into graduate school and continuing on for a PhD. Then I felt obligated to go into academia. I can't explain why, but I did. And I was in academia 15 years. I was a tenured full professor, but I began to slowly realize that much of the research that we were doing, we were not looking at solving the root causes of our problems. We were looking at research that treated symptoms. I call it putting a Band-Aid on a gushing wound. And I kept noticing that in spite of everything that was being developed and that I was a part of developing as a researcher, wasn't really moving the needle. Our soils were not getting healthier. They were becoming further and further degraded, and we were more and more reliant on inputs to produce our crops. Our livestock were not getting healthier. We were more and more reliant on supplements, pharmaceuticals, and so forth in our livestock. And I had to think back about the 6 generations of my family that didn't have all of that. They didn't have any of that. And somehow they supported multiple generations of family through all of those 6 generations. Okay, 4 generations pretty much consistently all the time. We're always on the farm. So multiple families supported by the farm. And how in the heck did they do it without all of these new technologies and new pharmaceuticals and new supplements and da-da-da-da-da? And I started realizing that we needed to look deeper at things and look at the root cause. How do we solve the root cause of our problems? And I quickly discovered that our universities and the funding streams weren't interested in that, because that didn't continue to develop funding streams and billions of dollars in sales and products to all of us farmers and ranchers. So I had a decision to make. I either stayed in academia until I could retire and keep on keeping on with the status quo, or I had to get out. There was no middle ground there. It didn't exist. So I made the decision to get out. That was 23 years ago in the year 2000. And I can say I haven't regretted that decision a single day. And we've done far, far more outside of academia than I ever could have done had I stayed in formal academia.
That's amazing. I love it. And actually, I forgot to start our facts end of webinar poll. So I'm going to put that up while we, oh, whoops. I've been sharing that for a long time. Sorry, I didn't mean to do that. But I'm going to let Alan answer the next question, and then I'm going to put up our next poll. And if you wouldn't mind answering that, I'd appreciate it. And maybe I'm not going to ask this next question first, because I actually really want to pay attention to your answer on that one. So I'm going to answer the next one. And hopefully we're going to get Alan back. Chris asked, what type of testing do you recommend to determine the health of your soil today? Your soil biology and pasture composition. And Alan and I were just talking about having him come back and do some more in-depth forages in different parts of the country. So we'll get more in depth with that, Chris, I promise, hopefully in February, March. But I'll let you go ahead and answer that, Alan, and I'll put up this poll.
Okay, so relative to testing for soil biology, we use the PLFA test. That stands for phospholipid fatty acid analysis. And what that does is that gives us several key parameters. It tells us our total living microbial biomass in the soil. It gives us a functional group diversity index. And then it breaks down that total living microbial biomass into the main microbial classifications. So we get total bacteria, gram-positive and gram-negative bacteria, total fungi, mycorrhizal and saprophytic fungi, and protozoa. And by looking at that, then we can quickly discern exactly what the biological status of our soils is and how we need to manage what are our strengths, what are our weaknesses. That's far, far, far more telling than a standard soil test. So there's basically 3 tests that we recommend all of our clients do, and we do ourselves on our farms. They are the Haney test, Haney, the PLFA test, and then a TND, and the TND stands for total nutrient digestion. And the reason we do that one is because a standard soil test only tells us in terms of soil mineral or nutrient status what is immediately plant available. Well, that's a small, small fraction of the total nutrients or minerals in my soil. The TND tells me the entire amount. It's a complete extraction. That's what I want to know, because then if I get my biology right in the soil, then my biology can make the totality of those elements or minerals available for plant uptake on a just-in-time, just-as-needed basis. That's why I want to know biology rather than pure soil chemistry. I'll let you in on a dirty little secret here, okay, from academia. Standard soil tests were developed originally to sell fertilizer. Plain and simple. And guess what? They do a fantastic job of that. They sell billions of dollars worth of fertilizer every year. And one of my jobs at the university, I used to have to interpret a standard soil test and provide recommendations to farmers and ranchers based on that. And never once did I ever get a soil test where I would say, oh, you're good, apply nothing, right? You always had a recommendation, NPK, lime, whatever. And so, again, that's all they do, folks. That's all they do. They just identify immediately plant available. And that's just a tiny fraction of what you need to know. And if all you're doing is relying on a standard soil test, I can guarantee you this: you are over applying fertilizer. You're over applying it, and you're harming yourself by doing that, both from a financial standpoint and from a soil health standpoint.
Mm-hmm. So I have a question for you. Where do you get your testing done? Because our state lab does not do that extensive testing.
So unfortunately, you're not going to get these done at your land-grant universities. And I used to be a part of that system, okay? When I was in academia, they don't offer this. Do they have the analytical equipment typically to do this somewhere at the university? Typically, yes. But do they offer this commercially for producers? No. Okay. Pretty much all you're going to get is a standard soil test at our land-grant universities, and that's unfortunate. And if we have land-grant folks on the line today, change that. Change it. Okay. Don't defend it because it's not defensible. Change it. And unfortunately, a lot of the commercial labs don't even do this, because most farmers and ranchers, what do they want? They want the standard soil test, right? Or maybe a sap test on the plants. So there's 2 principal labs in the U.S. that you can get this done at, the 3 tests combined. One is Regen Ag Lab, and you just go to regenaglab.com. The other is Ward Lab, WARD, and you can go to wardlab.com. And they both offer that suite of tests.
All right, thank you. Because that's always been the biggest problem is not being able to get affordable testing.
Yep.
So we've only got a couple of minutes left, but I do want to get to two more questions and then we'll say goodbye for the day. But because I think we could easily continue talking for quite a while. So, and this is what I really wanted to hear. So what would you mind sharing your standard answer that you give when customers ask you if you're using mRNA vaccines? Do you have a formal statement on your website?
So our answer is no, we are not. And we're not. Okay. And our customers have made it very clear to us, we do not want you using an mRNA vaccine. So we are very upfront about that we do not, and we will not, because again, we're going to give our customers what they want, period. End of story. Our customers want grass-fed, pasture-raised. We're going to give them that. Our customers want no antibiotics, no administered hormones. We're going to give them that. Our customers want no mRNA vaccine used. We're going to give them that. So that's our stance on our farms. And that's what we do in terms of the products we provide to our customers.
Okay, so I'm going to ask one more question. There's still quite a few good questions here, so I'm not playing favorites or anything. I'm just looking for one that might be able to answer relatively quickly. So Casey asks, are there any sources for funding for graduate research for what we, quote unquote, are trying to do? So I'm assuming that means regenerative ag.
So that there is more funding now being made available, and that's somewhat of a double-edged sword in that, even if the funding is available within the university system, you're still constrained by that university's administration and so forth and what they will allow you to do. Mm-hmm. It does work that way, folks. I lived it. And so all funding is constrained by a handful of things. One is what the funder wants. They want specific things, okay? And what your administrators and so forth want at the university that you're working at. So you have to recognize and understand that. Do the best you can, and but there is more funding being made available, and that's because there's more and more pressure being put upon the USDA, the federal government, and our land-grant universities to do regenerative research. I'll say one more thing on that. If you are a researcher and you're going to do regenerative research, do regenerative research and don't just call it that in name only. I can't tell you how many peer-reviewed articles I have read where they said they did regenerative research and they absolutely did not in any shape, form, or fashion do regenerative. Mm-hmm. They didn't even have a clue what it was. So if you're a university researcher, your PhD does not mean you understand regenerative. You first need to educate yourself on what regenerative is before you can adequately do regenerative research. So I'm going to throw out that challenge to all researchers and say, if you want to know what it is, come see us, because we eat it, breathe it, live it, and sleep it every day. And we can show you what it is.
I love it. The gauntlet has been put down. All right. I have a few housekeeping items to share before we sign off today. A recording of this webinar and the slides will be available hopefully this afternoon. It's going to depend on how long Zoom takes to download the recording. These documents will be archived on our website, and I will also email them to you. We also have some other good webinars coming up this autumn and winter, and I will share links to all our upcoming webinars and other opportunities for farmers and ranchers in the follow-up email. A sincere thank you to Alan. It was amazing. It's just blown away by how knowledgeable you are and how well you were able to field these questions. And finally, I want to thank everybody out in the audience. We just have the coolest people who sign up for these webinars, and they have such interesting things to say and such great questions. So kudos to you guys. Thank you so much. I hope that you had a good experience today, and stay in touch and connect again soon. Enjoy the rest of your day, and goodbye for now, everybody.